everyone, and welcome back to the Biochemistry YouTube channel. Let's jump right in. This is going to be one of many videos discussing the steps of nitrogen metabolism. In this video, I'll be talking about the urea cycle, or should I say the urea and ornithine cycle. But before we start, we must understand what steps even bring us to the urea cycle. Proteins are broken down in our body through a process called proteolysis. After breaking down proteins, we get amino acids. Amino acids go through a process called deamination to release two byproducts, ammonia and an alpha-keto acid. We get the ammonia from the amino group. The alpha-keto acid is the remainder of the structure after the ammonia is removed. However, ammonia is toxic to our bodies. We have to convert it to another molecule in our body called urea, which is the process that we care about today. Urea consists of two nitrogen atoms and one carbon dioxide. One nitrogen comes from aspartate, and the carbon dioxide comes from carbamyl phosphate. The aspartate is a molecule found in the urea cycle, and the carbamyl phosphate is formed in the urea cycle from ammonia and carbon dioxide. Let's take a general look at the cycle. It was formed of 5 reactions, with the 2 first steps happening in the mitochondria and the last 3 in the cytoplasm. Bear in mind that this entire cycle happens in the liver. In the first step, we have the ammonia and the carbon dioxide that join together to form a molecule called carbamyl phosphate, as I previously mentioned. You can guess the name of the enzyme as it synthesizes the carbamyl phosphate. Thus, the enzyme name is carbamyl phosphate synthetase. We are also using 2 molecules of ATP and turning them into 2 ADP molecules. N-acetylglutamate is an activator of this enzyme, meaning when there is more N-acetylglutamate, there is more production of carbamyl phosphate and more enzymatic activity when free amino acids are present. This makes sense, since when there are more amino acids, there is more ability to have deamination of amino acids, thus more production of ammonia, which we want to get rid of by producing carbamyl phosphate. In the second step, we combine carbamyl phosphate with ornithine to form another molecule called citrulline. This is done through an enzyme called ornithine transcarbamylase. Thus, citrulline and ornithine are both amino acids. You may be wondering why you haven't heard of them before. This is because they are not essential, meaning they are formed in the body and do not enter in the structure of proteins. We know that the first 2 reactions take place in the mitochondria, and the rest of the reactions take place in the cytoplasm. Thus, citrulline enters the cytoplasm in order for us to complete the urea cycle. The third reaction is somewhat similar to the second reaction. We have citrulline and aspartate, and they combine together to form arginosuccinate, which we synthesize and have great enzyme names. Thus, the enzyme that carries out this reaction is arginosuccinate synthetase. We said that urea is formed of 2 nitrogen atoms, and one nitrogen atom comes from aspartate and the other from carbamyl phosphate. The carbon dioxide also comes from the carbamyl phosphate. In this reaction, we are essentially splitting arginosuccinate for step 4. Thus, whenever we split a molecule, the enzyme ends with ase, and the enzyme itself is either referred to as arginosuccinase or arginosuccinate lyase. This splits the molecule into arginine and fumarate. What's great about this molecule is that it can also actually be transformed into glucose to form energy. The next great part is that the 5th reaction is very similar to the 4th. It is the splitting of arginine to form 2 molecules. The first is the end product of the cycle, urea, and the second one is ornithine, which can enter into another cycle. The 5th reaction takes place in the cytoplasm, which means that the urea is produced in the cytoplasm, and it is transported through the blood in your body, so it can reach the kidneys and be excreted in urine. Ornithine is also produced in the cytoplasm. I'd like to show you this diagram because I think it makes it look a little bit more clear. Since the second reaction takes place in the mitochondria, ornithine can actually be shuttled back into the mitochondria to repeat this cycle so that more urea is secreted from the body and that ammonia is converted into a safe form that can leave. But that's about it. Make sure to watch the next video on T3 and T4 metabolism, as well as purine and pyrimidine biosynthesis.